Welcome to BioPharm International's podcast, Critical Aspects of Antibody-Drug Conjugates, Structural Characterization and Analysis. Brought to you by Eurofins Lancaster Laboratories, a global leader in laboratory services supporting the needs of more than 800 biopharmaceutical companies through a full scope of laboratory expertise, ranging from method development and characterization services through marketed stability release. And now, here's your host for this podcast, Angie Draculich.
Hello, this is Angie Draculich, Editorial Director of BioPharm International Magazine, and I'm here today with Lancaster Laboratories Biopharmaceutical Services Manager, Dr. Robert Duff, and Biochemistry Principal Scientist, Dr. Liza M. Danon-Leon. Today we're addressing critical aspects of antibody-drug conjugates, structural characterization, and analysis. Thank you both for joining me today. Bob, can you get us started by explaining to listeners exactly what antibody-drug conjugates, or ADCs, are?
Antibody-drug conjugates are a class of molecules that are rationally designed, and they're formed through chemical linkages of a small molecule, cytotoxin, a drug that is currently on the market or one in discovery, linkage to a monoclonal antibody, and they have complex and heterogeneous structures, more complex than the antibody alone.
And Liza, what are the benefits of using ADCs in drug development over other approaches?
Well, the targeted drug delivery approach of ADCs minimizes the exposure of non-target cells to cytotoxic agents, thereby reducing potential side effects of the treatment. This kind of approach have implications on the lowering of the dosage and lessening the frequency of chemotherapy, which pretty much affects the quality of life of the patient.
And are there any challenges associated with developing antibody-drug conjugate products?
There are a lot of challenges right now. The main challenges associated with ADC drug development revolve around the stability and heterogeneity of the ADC produced and how it affects the drug product's efficacy and potency. Understanding this relationship will help in the identification and development of a long list of assays that can accurately and precisely characterize the ADC drug development both in vitro and in vivo. These are just some of the concerns that will have to be addressed and pose as the main challenge in developing these kinds of drug products.
Great, thank you. And Bob, just to come back to you for a bit, I wanted to talk a little bit about the regulatory issues. Where do ADCs stand with regard to FDA approvals?
Yes, there are two ADCs that were approved by the FDA. The first one, Mylotarg, and the second one, most recently, is Adcetris. Mylotarg was recently withdrawn from the market due to linker issues of stability. There are other candidates in the pipeline of various pharmaceutical companies. About 7 or 8 pharmaceutical companies are currently pushing these molecules through their clinical pipeline from phase 1 through phase 3.
Great, thank you. And what regulatory requirements are involved and what procedures should be taken to accommodate regulatory agencies?
These regulatory requirements are quite complex since these molecules are conjugates and they have various offices within the FDA, the Office of New Drug Quality Assessment, the Office of Biological Products, the Office of Pharmaceutical Sciences, and the Division of Monoclonal Antibodies, or the DMA. The problems that have occurred have been in the designation of the starting materials and the drug substance as per the ICH guidelines Q7 and Q11. Basically, the challenges within these agencies is governed by the designation of the materials and the passing of the project from one agency from, for example, the ONDQA to the Division of Monoclonal Antibodies after the conjugate has been produced.
Great, thanks for that information. Bob, just one more question. I was wondering if you could tell listeners how they can learn more about ADCs.
Well, Lancaster Labs will be hosting a webinar on September 25th on the critical aspects of antibody-drug conjugates. A more descriptive and complete discussion will be held on the challenges of analytical method development that is required for physicochemical characterization, as well as regulatory issues that each of our clients may face. To register, visit LancasterLabsFarm.com.
Great. Thank you both so much for your insight on antibody-drug conjugates.
You've been listening to BioPharm International's podcast, Critical Aspects of Antibody-Drug Conjugates, Structural Characterization and Analysis, brought to you by Eurofins Lancaster Laboratories. To find out more, please visit them on the web at LancasterLabsFarm.com.